Hey everyone, and welcome back. It's great to have you with us for another special session from the Dr. Cool Cat team. Today, we are on a mission. We're going to crack the GED genetics code. If you've ever felt like those science questions are written in a totally different language, trust me, you are in exactly the right place. We're going to turn that confusion into confidence and help you score some serious points on test day.
Okay, let's just be real for a second. You're taking the GED Science test, you're in the zone, everything's going great, and then bam! You hit a question about dominant traits, recessive genes, and this weird little grid thing called a Punnett square. If that makes your stomach do a little flip, you are not alone. It's kind of designed to look scary.
But here's the secret, okay? These questions, they are some of the most predictable, most formulaic questions on the entire test. And today, I'm going to give you a simple, foolproof method to get them right every single time. We're not just going to face that panic, we're going to flip it and turn it into some of the easiest points you are going to get.
First things first. Before we can play the game, we've got to know the players, right? To really master genetics, we have to get the core vocabulary down. Think of it just like that, learning the key pieces on a chessboard. Once you know what they are and what they do, everything else just starts to make sense.
So let's start with this word allele. It sounds complicated, but it's not. Just think of a gene as a general instruction, like hair color. Well, the allele is the specific version of that instruction, like brown hair or blonde hair. It's that simple, and you get one version from each of your parents, which creates your own unique genetic combo.
Next up, we have dominant alleles. You can kind of think of these as the loud or bossy ones. We always write them with a capital letter. And here's a great way to remember it. A dominant allele is like a super bright LED light. Even if you only have one, it's so powerful it's the only one you're going to see.
And on the other side, you have the recessive allele, which we show with a lowercase letter. These are way quieter. Think of them like little candles. Now, if you have one bright LED light and one tiny candle, you're only going to notice the LED, right? But if you get two candles together, well, then their light finally gets to shine through. And that's exactly why you need two copies of a recessive allele for that trait to actually show up.
Okay, this brings us to a really crucial difference. Genotype versus phenotype. The genotype is the actual genetic code, the combination of letters, like FF. The phenotype, on the other hand, is the physical thing you can actually see with your eyes, like having free earlobes. Here's an easy way to remember it. Geno is the code, pheno is the photo.
And the last two key terms you absolutely need to know. Homozygous means you have two of the same allele. The prefix homo means same. So that's either two dominant alleles like capital F, or two recessive ones, lowercase f, lowercase f. Heterozygous means you have two different alleles. Hetero means different. So that's always one of each, a capital F and a lowercase f.
All right, you've got the vocabulary down, you know the players, now you need the game plan. So I'm going to share a super simple acronym that is going to be your secret weapon for every single genetics problem on the GED. It's a step-by-step routine that basically guarantees the right answer. It's called gamete.
So here's how this works. G is for give letters to the traits, A is for assign the parents' genotypes, M is for make the gametes, that's what each parent can pass down, E is for enter those gametes into the Punnett square, and the final piece, TE, is for tally your results and express the answer in whatever form they ask for. That's it. That's the playbook.
Okay, enough talk, right? Let's run some plays. It is time for a little practice scrimmage to see our gamete routine in action. We're going to walk through a classic GED-style problem, and I'll even point out a super common trap you've got to watch out for.
Okay, here's our scenario. The trait is earlobes, and we're told free earlobes are dominant. So we've got one parent with attached earlobes. Now, since that's a recessive trait, their genotype has to be lowercase f, lowercase f. The other parent is a carrier, which is just a fancy word for heterozygous, so they have one of each, capital F, lowercase f. See that? We just did steps G and A of our routine without even trying.
So now we just follow the playbook. Next up, M and E. For M, we make the gametes. The FF parent can only pass on a lowercase f. That's it. The FF parent, though, could pass on a capital F or a lowercase f. For E, we enter them in the square. We put one parent's options across the top and the others down the side. Then you just fill in the boxes, combining the letters from the top and the side. It's like a little puzzle.
And now for the final steps, tally and express. We just look inside our four boxes. What do we see?
We've got 2 boxes that say FF and 2 boxes that say FF. So what does that mean? Well, FF means they have free earlobes, and FF means attached. The result is a perfect 50-50 split. There's a 50% chance the offspring will have free earlobes, and a 50% chance they'll have attached.
Now, hold on, because this brings us to a major trap. It is so easy to think that dominant also means more common or more likely, but that is not always true. Dominant just describes how a single allele acts. Your job on the test is to completely ignore any assumptions and just trust the math you did in the Punnett square. The square never lies.
Feeling pretty good about that? I hope so. So let's level up. Let's try an advanced pro-level move. Sometimes the GED will throw a curveball with something called a sex-linked trait. Don't panic. The logic is exactly the same. We just have to pay a little extra attention to the X and Y chromosomes.
Okay, so here's the new setup. We're looking at a trait like colorblindness, which lives on the X chromosome. So we have a mother who is a carrier. That means she has 1 normal X chromosome and 1 affected X chromosome. The father has normal vision, so his 1X is normal, and then of course he has a Y chromosome.
We fill this out just like we did before. The father's 2 possible gametes, his X and his Y, go across the top. The mother's 2 different X's go down the side. Now, as we combine them, watch what happens. Daughters, who are XX, always get an X from both parents. But sons, who are XY, always get their X from their mom and the Y from their dad. That is the absolute key to these problems.
Now, reading the results here is critical. Let's do it carefully. Look at the 2 daughter boxes on the left. Both of them result in normal vision, so 0% of the daughters are colorblind. Now look at the 2 son boxes on the right. 1 son is normal, 1 is colorblind. So that means 50% of the sons will be colorblind. And this right here is the pro-level trap. The GED loves to get you with the wording. If the question asks, what percentage of the sons are affected, the answer is 50%. But if it asks, what percentage of the children are affected, well, that's only 1 out of the 4 boxes, so the answer is 25%. You have to read the question with a magnifying glass to see exactly who they're asking about.
Whew. Okay. We've covered a lot, so let's boil it all down into one simple final checklist that you can just keep in your head on test day. This is your go-to strategy, your game plan.
Just remember, game it. G, give the letters. A, assign the parents. M, make the gametes. E, enter them in the square. T, tally up the results. And E, express the answer. If you follow those 6 steps every single time, you simply cannot go wrong.
All right, your turn. Time for you to be the expert. Here is a classic GED problem for you. The ability to roll your tongue, we'll use R, is dominant. So we have 1 parent who is heterozygous, RR, and 1 who is a non-roller, RR. Use the gamete method we just learned and figure out what percentage of their children are predicted to be non-rollers. Go ahead and pause here if you need a minute to work it out on paper.
You absolutely have this. You have all the tools you need to solve that problem. Work it out and then drop your answer down in the comments below. And tell me, which step of the gamete routine was the most helpful for you? You have officially completed your genetics training. You are ready. Thanks so much for joining us. And hey, if this helped clear things up, do us a favor and like this explainer and subscribe for more GED strategies from all of us here on Dr. Cool Cat's team. Now go ace that test.